Welcome student in our next lecture about our discussion of secondary growth and in this we will talk about a different type of secondary growth, that is periderm, which is formed in stem as well as in root. Now the secondary growth which we have discussed was in the vascular region, okay, formed by vascular cambium. Now this we have already discussed that in this inside the stele, okay, it is known as intrastellar also because this cambium arises inside the stele, okay. You have vascular bundles in the stem in the form of rings, and then you have intrafascicular cambium and interfascicular cambium. They will join to form a ring like structure and they will give out phloem on the outer side and xylem on the inner side. So that all we have discussed. With this there will be increase in the diameter of the tissues which are present in the vascular region, okay, and a large amount of xylem will be formed as compared to phloem. So that all we have discussed, and a small amount of phloem will be there, because the activity of cambium is as such that it will cut more xylem tissues as compared to phloem. So more xylem is there. Another reason is that as the xylem is increasing in diameter towards the outer region, most of the primary phloem as well as the older secondary phloem, they all get crushed. So their layer is also reduced as compared to this xylem, secondary xylem, okay. Now see that also I have told you before that when the tissue is increasing in diameter from inside, okay, the outer region should also expand to accommodate these expanding tissue, okay. So definitely there will be an expansion of the outer layer like cortex or everything after the vascular cambium. So in that also there will be an expansion. So that expansion will be taken care by another cambium which is known as cork cambium, okay. So this cork cambium will finally give rise to a structure which is known as periderm, okay. So outer initially in the outer layer you will find epidermis, but now it will be replaced by another protective layer which is known as periderm. And it will, this periderm will also replace the cuticle. Like in the primary tissues, the epidermis and cuticle, they are the protective layers and they are not very thick layers, okay. Mostly the epidermis is living cell only, and cuticle, the thickness of the cuticle will help in determining the level of protection. So that all will now be replaced by a thick layer. You can say that it is not living also, and that will provide protection from the outer side, and it is found in dicot and gymnosperm where secondary growth will occur. So it will be there in stem as well as root. So both root and dicot stem both will undergo this type of secondary division. Okay. Now this periderm which is formed, it is formed by cork cambium. Now this cork cambium, how this cork cambium will arise? See, now I have told you before that in the cortex and endodermis, pericycle, lot of parenchymatous tissues are there, and one of the important function of parenchyma is also that it can undergo dedifferentiation and perform meristematic activity. See, this is very important, that it's not that only the vascular cambium has meristematic activity or the or the apical tissues have, it's also the parenchymatous tissue. Wherever it is required, these parenchymatous tissue will undergo dedifferentiation and will perform meristematic activity. So in the entire ground tissue, lot of parenchyma is there. It is there in the epidermis, it is there in the cortex, pericycle, endodermis. So lot of parenchyma is there. So in those regions, we call it, suppose in the cortical region, okay, the parenchymatous tissue which is present, that will start behaving like a meristematic cell, okay. They will start behaving, and this meristematic region will be known as cork cambium. Now this cambium will behave almost in a similar way as that of vascular cambium, cutting tissues on either side, but definitely they will not be a xylem or a phloem tissue. Okay. Now this core cambium is also known as phellogen. See, don't get confused in all these names, because many a times in exams a different nomenclature is by questions could be asked by their other names also. So it's important that you should remember all the names. So core cambium is also known as phellogen. Okay, it will cut phellum on the outer side and phelloderm on the inner side. Now phellum is also known as cork, whereas phelloderm is also known as secondary cortex. Okay. So all these three layers together are known as periderm. Okay. So this periderm is the outer protective tissue. Okay. It is made up of phellogen, phellum and phelloderm. Now this is just the figure which shows you that how in the center you will have a small amount of pith and then a large patch of xylem, then a cambium in the center and phloem, and from the outside you will see that phellogen is giving rise phelloderm on the inner side and cork or phellum on the outer side. See, epidermis it is shown here, but later that epidermis will be crushed and it will be ruptured. So you may not find definitely epidermis on the older tissues which have developed a mature periderm. Okay. This figure also shows certain tissues that we will discuss later, but here you can find out another name which is given here is bark. See, commonly you call the outer layer of a woody plant as bark also. Okay, and that bark is you would have noticed that you can easily take out that bark. Okay, so bark is also that outer hard tissues which forms the protective layer. However, it might be slightly different, although the periderm is also a composition of bark, but we give the term bark to all those tissues which are lying after vascular cambium. Okay, so that also we will discuss afterwards. Now coming to core cambium. See, core cambium is a lateral meristem. Okay, it is a lateral meristem. Okay, that means it is also found on the lateral side as cambium. Okay, so cambium was there, suppose a cylinder of cambium is there and outer side you have this core cambium. And here cambium was there, vascular cambium. Okay, better we call it as vascular cambium. Now definitely they are dividing cells. Okay, it is a temporary meristem. See, core cambium is not found from the origin of the plant, like it is not found in the primary tissues. When the tissue is dividing only by primary growth, you won't find core cambium. So definitely core cambium is a secondary meristem. So that also we have discussed in previous classes. See, it's secondary meristem, whereas vascular cambium was a primary meristem because it originates after the growth of a plant. Now phellogen originate by de-differentiation of existing cells. So what could be the cells which will give rise to this phellogen or the core cambium? So definitely they will be epidermis can give rise to core cambium. Okay. Then you have cortex, it could also originate from cortex. It could also originate from pericycle, even endodermis also. Then it can originate from secondary phloem. See, all those tissues contain parenchymatous cell type. Many of the cell types present in these layers have parenchyma. So they can perform meristematic activity. Now the phellogen is basically one cell layer thick. See, as in case of vascular cambium also, this phellogen is also one cell layer thick. Okay, it's not multicellular. Okay, only this is one cell layer thick, and as in case of vascular cambium also, it was giving rise to phloem on the outer side and secondary xylem on the inner side. Here also it will give rise to cork on the outer side and secondary cortex on the inner side. So, in this case also multiple layers of cork will be produced. See, one cell will divide into two, the one will form cork and the other will form, will remain meristematic, again it will divide, the other will form the secondary cortex or phalloderm, and the other one will remain as meristematic, and the division will be periclinal. See, in the vascular cambium also the division was periclinal, because in periclinal division the increase in diameter occurs. See, the tissues are coming out in this outer direction and the inner direction. So, in that way diameter is increasing. So, this type of division is known as periclinal, whereas anticlinal division that we have discussed, that this will just increase the number of layers. So actually periclinal division will actually increase the volume, and this will increase the number of cells. So, the diameter of the cork cambium will increase, okay. It will increase the surface area of the cork cambium, whereas the periclinal division will increase the volume, okay, of the inner side and of the outer side, as we have seen in vascular cambium. And phallogen is bifacial in stems and unifacial in root. See, bifacial because it will give rise to tissue on either side, on one face as well as on the other side, whereas sometimes in root it might give rise to tissue on one side also, sometimes that case may also arise. Now, there you can categorize phallogen on the basis of from where they are originating, okay. A phallogen could be said to be superficial if it is originating from epidermis or a few layers below epidermis, might be few layers in the cortical region, you can say, or near the surface of epidermis. Now, deep seated phallogens will definitely arise from the interior of the stem. So, definitely they can arise from secondary, they can arise from cortex also, okay, some superficial layers of cortex, secondary phloem, okay, pericycle. So, these layers can give rise to deep seated phallogen, whereas in root it is always observed that deep seated periderm definitely arises from the pericycle. So, that also we have discussed several times that pericycle has lot of meristematic activity and it helps in formation of the periderm, however it also in root it helps in formation of the lateral roots also. Now, the region from where actually this phallogen or the cork cambium is arising, that is also important. Suppose it is arising on the, suppose this epidermis has changed its activity and it has started giving rise to cork and secondary cortex or the phalloderm on the inner side and phloem on the outer side. So, you can see here that on the inner side we will have cortex, the inner layer you will have cortex, okay. Then you will have secondary phloem, okay, because pericycle and endodermis mostly they will be crushed. So, they are just single layers or some patches of pericycle you can find, and then you have xylem. So, many of the tissues will be found on the lower layers of these this cork cambium. And suppose if you have secondary xylem and you have secondary phloem, suppose the cork cambium is arising here, okay. So, definitely it will give rise to cork and phalloderm on the inner side. So, most of the tissues outside secondary phloem will become dead because they will be pushed outside and most of the tissues will be converted into cork, that we will discuss that cork is actually impervious to water. So, most of the tissues which are outside to this cork will become dead. So, you can notice here that depending on the position from where this cork cambium is coming out, the thickness of the periderm or whatever later we will see that the thickness of the bark, that is all is decided. So you would have noticed also that in that bark is also variable in different plant types. In some very woody and thick or big plants, you can see a very thick bark is coming out, whereas in small plants, although they have secondary growth, but the bark is not very thick, and when you are cutting the sections, you will also see well developed cortical layers, then pericycle, endodermis, and along with that a very thin bark is there. But in many woody tissues, even bark could be is very thick, and you can even take out that bark, and lot of many products are made from that bark. Okay, so that where that say cork cambium is coming out, that is also very important deciding factor. Now phellum, that is cork. See, this cork is very important part of periderm. Okay. So cork, the word you would have heard when Robert Hooke have. So basically corks, what are corks? They are also cells only, but they are dead cells. So you all would have heard about Robert Hooke identifying the cork cells, that from the beginning the cell term was given by Robert Hooke, and he identified the dead cells of cork, and he thought that these are like chambers and gave the name to them as cells afterwards, but living cells were also identified. So like this somewhat dead chambers they were found with walls, okay, covering of wall structures. So when we observe cork, we will find that they are radially arranged into, they are radial brick like columns, okay. They are actually radially arranged cells which form brick like columns, okay, with no intercellular spaces, almost they are tightly packed. You can see here also that these cork cells are tightly packed, okay, and this is the cross section, this is the TS, and in the longitudinal section you can say that, see here that they are radially arranged, okay, with very small or almost no intercellular spaces. An important thing is that the cell wall of cork contains suberin. Okay, suberin is also a chemical which is impervious to water. See, that also we have discussed that cell wall could be deposited by various other substances, like lignification was there. So lignin deposition was there, and in that also cell was actually dying after lignin deposition. In suberin also it is a hydrophobic compound which deposits on the cell, and afterwards cell at maturity they die. So just remember here that you do not have lignin deposition, you have suberin deposition. Now the lumens might be empty, they might be filled with air, okay, and many other substances are also reported to be filled in those lumens, like tannins and resins. See, these substances, many things from bark also you would have noticed that many things are coming out from the bark. So from where they have arisen? In the bark cells also some tannins are there, some resins deposition like terpenes are there. So all these substances might be deposited in the cork cells also. Here is also you can see a figure where few cork layers are getting formed, and this will be the cork cambium, and this is the phloderm, and this is the phellum, and this is the phloem. See, here also you can see that single cork layer is there, which is giving rise to more single cork cambium is there, which is giving rise to more cork layers as compared to the phloderm. So always more cork is formed. Like in vascular cambium, more xylem was formed, similarly more cork is formed. Now phloderm, see, phloderm is actually not produced in every species. See, cork is definitely produced by cork cambium, okay, but phloderm is also there, but it is not commonly found in every plant species. Depending on the requirement, the phloderm can vary, or their thickness can also vary, and whether many a times it is not even produced. It is also known as secondary cortex because of its location, because it is located near to the cortex, so it is also known as secondary cortex. Now phloderm cells are also aligned in radial fashion, okay, they are also aligned in radial fashion. okay, and these cells are larger and they are also parenchymatous. See, important thing is that cork cells were dead cells, okay, but they are parenchymatous, so they are living cells at maturity as compared to cork are living, okay. So they also store starch. So because they are parenchymatous, one of the important function of parenchyma is storage. So these phalloderm can also store starch, and this we have discussed that phallogen generates more phloem as compared to phalloderm. Now, if we draw the structure of all these layers, suppose you have a pith, okay. Inside this pith, you will have primary xylem. See, primary xylem won't be continuous or because in the beginning actually vascular bundles, if we talk about stem also, they are in discrete patches, okay. They will become continuous only after secondary growth. So initially whatever the primary xylem is there, they will be in the discrete patches, that will be followed by a big layer of secondary xylem, okay. Then you have a smaller patch of secondary phloem, in which you can see primary phloem crushed, okay, crushed primary phloem, okay. Then you have phalloderm, then okay, first let's try to label it. See, pith will be there, that will also be filled with parenchymatous layer, and in older tissue that might be sclerified also. This is primary xylem, this is secondary xylem, then you have secondary phloem, then primary phloem, okay. Then you have phalloderm, this will be phallogen, and this will be phloem or cork. Okay, and above you will find some crushed patches of epidermis, which may be visible or may not be. We can assume that some crushed patches of epidermis will be there, that will be crushed. So in this way you will find different layers, okay. Then in the periderm, you will also find a structure which is known as lenticels. See, like in leaves and in herbaceous stems also, like basically on leaves, we find pores which are meant for gaseous exchange. Similarly, in stem also, which has developed periderm, see, because that layer, cork layer is completely impervious, so some sort of pores should also be there which could help in gaseous exchange. So these are known as lenticels. See, you would have seen some spots on fruits also which develop periderm, on stem also some spots are there, white colored spots. So they are basically lenticels, okay. So what are lenticels? They are actually loosely arranged cells which are found above in the cork layer actually. See, this is the cork layer. In the cork layer you will find some loosely arranged cells, okay. So they are, and they just open like a pore, and these are filled with loosely arranged cells which are known as lenticels. So these pores help in gas exchange, okay. It is found in roots, stems and fruits also. They are the cork cambium which is below these lenticels are very active, and they are known, their meaning, they have been taken from a Latin word known as lens. Okay, they are also sometimes very important characteristic to identify species, because their structures or how they are present in the species can vary. Then now coming to very important concept, you would have heard of the name bark. Okay, see, it's a non-technical term which is given to all the corky tissues which are present outside of a plant, actually outside of a plant vascular cambium, means anything outside vascular cambium is known as bark. Okay, anything outside vascular cambium is known as bark. So it will definitely include, see, suppose this is the vascular cambium, this is the xylem, okay, this is the secondary xylem, and this is the cambium, the vascular cambium. After this you will have a small amount of phloem, secondary phloem, then you will have phellogen, then you will have cork cambium, then cork is there. Okay, so all the things, this is secondary xylem and this is secondary phloem. So everything outside this vascular cambium is known as bark. Okay, so because as xylem, because more amount of definitely xylem is found in woody tissue, and in woody tissue also you will find a well developed bark also. So this, when it is pushing, most of the xylem is pushing, most of the cells are pushing towards the outside, and when they are moving them, they will actually be replaced by dead tissues. They will reach to the surface and they will be formed into periderm, cork, cambium, and so many of the tissues will actually they will shrink and they will be compressed, they will actually die and they will form the portion of bark. However, bark also is made up of two, you can divide them into two portions, means everything from vascular cambium to cork cambium is the inner bark. In this you will have actually secondary phloem and secondary cortex, okay, that is the phelloderm and the secondary phloem. So it will be made up of somewhat living cells. So you can say that the inner bark is living cell, but this will also be pushed on towards the outer side, and they will be replaced by more dead cells, because the cells will eventually die when they are moving to the outer side. They will not have the reach to water and nutrients. They will not reach those tissues. So finally they will die, and they will be replaced and containing cellulose and lignin molecules. Now outer bark is from cork cambium to everything to the outside. You have cork cambium. After cork cambium, everything outside will form the outer bark. So in that also basically you have cork cambium, and however, if it is arising from the, suppose it is arising, this cork cambium is arising in the secondary phloem region. So then everything outside to this cork cambium will be the outer bark. So depends where the cork cambium is arising. However, the inner bark will contain living components and the outer bark contains mostly the non-living. Rhytodyme is a name which is also given to outer bark. We call them as rhytodyme also. It is not wood. See, it appears like wood. You will think that it appears like wood, but it is not wood, because wood is what? It is secondary xylem. But these barks actually, they do not contain secondary xylem at all. They are dead cells. Outer bark is mostly made up of dead cell, and it keeps on adding each year. But however, their increase is quite less when we compare to the xylem rings as of spring and autumn rings. So their increase in diameter is less, and but they will, if time is there, depending on the age of the plant, they will become quite thick. Now this bark, they contain lot of living components. They contain sieve cells and sieve tube as component of phloem. They contain fibers and parenchyma, which are all there in the cortex, and they all have been converted into bark now. Resin canals, then cellulose, hemicellulose are also present. They contain lignin, because all these cells where they are present. Okay, so they are now have been converted into bark. Now, but here they also form rings, but rings are not very prominent. We cannot distinguish between them. However, there is an annual increase also. Lot of medicinal chemicals are also extracted from bark, like quinine. You would have heard, it is an anti-malarial drug which is obtained from cinchona bark. Then lot of important resin, latex, tannins is also obtained from these bark. These barks are also used as board in making a different type of material, like the wall covering, many other material also. These bark are used. Many spices are also obtained from bark, like cinnamon, which you have heard of. These are obtained from bark of a tree, basically from the inner bark. Okay. Now, very important component of bark is cork. See, it is commercially also very important, and it is obtained from outer bark. Basically, the cork obtained from oak tree is commercially very important, and this cork is harvested from that tree after every around 9 years. Okay, time is given because it will require time to accumulate in that tree, and after every 9 years, you can see here how by a push or a pressure, these outer layer could extract, because they are not living, they are not completely attached with that attachment is not there as it is with the living cells. See, this cork is very important because it is impermeable, it is buoyant, elastic in nature, it has fire retardant properties, so it is made up of into lot of commercial product like cork stoppers you would have definitely seen, then in making flows and many other household things, these cork is used. So, this is all about the outer secondary growth. Now, I would like you all to just examine, in this case also you try to examine the trees nearby you and try to identify whether these trees have bark and how thick those barks are, and also try to identify if you in your house, if you have any product which is made up of bark. So, okay, so maybe I hope so some understanding definitely would have got, and try to identify these structures around you. So, for today, thank you.